Hello everyone. Today we are going to learn in vitro methods to study antibacterial properties of nanomaterials. In today's lecture, we are going to learn various techniques to assess antibacterial properties of nanomaterials, antibacterial mechanism of nanomaterials, and at the end of the lecture, we will also have a practical demonstration to assess the antibacterial properties of nanomaterials. Before I talk about the various methods for assessing the antibacterial properties of nanomaterials, let us briefly learn what is the need for developing the nanomaterials with antibacterial property. Bacterial infections are a major problem as they can lead to serious illness and even death. The bacterial infections can be controlled by use of antibiotics, but the extensive antibiotic usage will lead to multi-drug resistant bacterial strains. To overcome that, we need new materials with antimicrobial property. In search of novel materials, researchers found that nanomaterials exhibit significant antibacterial activity and they are less likely to promote bacterial resistance. So, these are the advantages of nanomaterials. That is the reason nanomaterials with antimicrobial properties are gaining much importance in the biomedical field. Let us learn about various methods to assess the antibacterial properties of nanomaterials. The first method is the turbidity assay. By using this assay, we can identify the MIC and MBC. MIC means minimum inhibitory concentration, that is the lowest concentration of an antimicrobial agent that will inhibit the growth of microorganism, and MBC means that is the minimum bactericidal concentration, that is the lowest concentration of an antimicrobial agent that will prevent the growth of an microorganism. That means MIC concentration, if you are using this MIC concentration, that will inhibit the growth of bacteria. If you are using this MBC concentration, that will kill the bacteria. To detect the MIC and MBC, we have to inoculate 300 microliter of 0.5 OD bacteria into 3 ml of bacterial growth medium. Then we have to add various concentration of nanomaterials and incubate overnight in the incubator. For example, if you have made some new nanomaterials and you want to study the antibacterial property of the new nanomaterial, we do not know at what concentration it will inhibit or at what concentration it will kill the bacteria. So, for example, in this case we can add, like for example, 2 microgram, 4 microgram, 6 and 8 microgram and 10 microgram and 12 microgram. So, once you add various concentration of your nanomaterials, then you can incubate for overnight. After overnight incubation, you can see the turbidity. Turbidity means the bacteria is grown. In the test tube, that is the control, that means it is untreated, where you did not add any nanomaterial. There is no nanomaterial, so here the bacterial growth is complete, you get the complete turbidity. Whereas you added the nanoparticle at the low concentration like 2 microgram or 4 microgram, still you are able to see the turbidity. That means the bacteria has grown in these test tubes. You can observe that in these 3 test tubes there is no bacterial growth. From this we have to identify which is MIC and which is MBC. For that, what we have to do is we have to re-inoculate from this test tube to a new test tube with 3 ml of bacterial growth medium. We have to inoculate 300 microliter from this test tube, and after incubating for overnight, you can see that there is a growth in the 8 and 10 microgram concentration, and there is no growth in the 12 microgram concentration nanoparticle. It shows that this 8 and 10 microgram is the MIC, that means it is able to inhibit the growth of bacteria. That is why when we re-inoculate into the fresh test tube, it is able to regrow again. Whereas in case of this 12 microgram, there is no growth. That means this is the bactericidal concentration, that is the killing concentration. And you can also do the spread plate method by taking 100 microliter from each test tube and incubate it overnight to observe the bacterial colonies. You can take 100 microliter from each test tube and you can do the spread plate technique, and after overnight incubation you can see the colonies growth, and you can also count the number of colonies. You can see here in the control there are a lot of colonies, and in case of MAC also you can see some few colonies are there, but in case of MBC there is no bacterial colonies. It confirms the which is MAC concentration and which is the MBC concentration. From this data you can understand this is a control, that means we did not add any nanomaterials, only bacteria we added. So, it is the complete growth of bacteria. You can observe the complete growth of bacteria, that is the turbid, complete turbid is there. And in case of different concentration of nanomaterial, you can see here there is some growth, but in these two concentrations there is no growth. And by using this kind of re-inoculation as well as by spread plate method, we can identify which is MIC and which is MBC. And here we used bacteria, that is a GFP E. coli. GFP means green fluorescent protein expressing recombinant E. coli bacteria, and it was treated with different concentration of AgZNO nanocomposites. We can also identify the MIC and MBC by optical density measurement, that is a OD. You can also see that when you measure the OD at 600 nanometer, you can see that the bacterial growth is going down with respect to the concentration. And whenever we perform antibacterial studies, we have to use at least 1 gram positive bacteria. In this case, we used Staphylococcus aureus, and at least 1 gram negative bacteria, that is in this case we use E. coli. So, we have to use 1 gram positive and 1 gram negative bacteria to understand whether our nanomaterial is having broad spectrum of antibacterial activity against both gram positive as well as gram negative bacteria. Also, this MAC and MBC also varies with respect to the bacteria. Okay. You can also see that in SRES this MAC concentration is different, and in the E. coli the MAC concentration and MBC concentration is different. So, we have to use the right concentration to restrict the complete bacterial growth, and here you can see that N equal to 3, that means this experiment was done 3 times, and the data represented here, these are the average of that, or it can be a triplicate samples, and this error bar represents the standard deviation of the data. So, you can observe here for SRES the same nanocomposite, it is giving 60 microgram as MAC, for E. coli it is giving a 550. So, depends on the nanomaterial and depends on the bacteria, this MAC and MBC will vary. And we can also do the time dependent study to understand at what time point the nanomaterial is killing the bacteria, and in this case we used a silver nanoparticle, and the silver nanoparticle is made using sodium borohydride as a reducing agent. So, to confirm that the bacterial death is due to silver nanoparticle and not due to the reducing agent, we have to use sodium borohydride as another control. One is the negative control where you are not adding any nanomaterial. In this case, you can see that bacterial growth is proper and it is growing more than 12 hours. And similarly, when you are adding the reducing agent, that is sodium borohydride, there also there is no bacterial death, it is equal to control, it is growing nicely. And when you are using the different concentration of silver nanoparticle, you can see that with respect to the concentration and with respect to the time, you can see the bacterial growth is going down. If you are using low concentration, the bacterial growth is there, but it is less compared to the control. Whereas in this case, you can see here the bacterial growth is going down when you are using the optimum concentration, there is no bacterial growth. So, we can do the time dependent study as well as the concentration dependent study for understanding the antibacterial properties of nanomaterials. So, you can see here in control that is untreated, there is no effect, and when you are using only reducing agent, that is the sodium borohydride, there is no effect. And nanoparticle treated cells, as I told earlier, with respect to the concentration, the bacterial growth is going down. And we can also do the colony counting method, as I told earlier. From the with respect to different time point, you can take the bacterial culture 100 microliter and do the spread plate method, then you can count the number of colonies and you can plot it. You can see that with respect to the time, the number of bacterial colonies are going down, and also with respect to the concentration. You can see that at a 3-hour time point there is some bacterial growth, but at the time of 6-hour and 12-hour there is no bacterial growth. So, by using this colony counting method also we can understand the antibacterial properties of nanomaterials. And as I told you, like in this study we used bacteria, that is a green fluorescent protein expressing E. coli. So, this is a kind of recombinant bacteria and it emits green fluorescence under UV light, and this is a control is untreated. That means no nanoparticle was added to this bacteria. You can see here the intact bacteria. This is a E. coli bacteria. This is a rod shaped bacteria. You can see the intact rod shape under the microscope. Whereas the silver nanoparticle treated, you can see that the bacteria is damaged. It is undergoing cell death. And the advantage of this green fluorescent protein expressing E. coli is we do not have to add any additional fluorescent dye or stain to see the bacteria under the microscope. It has its own GFP, and under the UV light we can easily observe the GFP expression. So, that is the advantage of this GFP E. coli. And here also we can do the time dependent and concentration dependent study. You can see here this control is untreated. With respect to time, the number of bacteria is going high. You can see at 0 hour the number of bacteria is less, whereas at 12 hour you can see the number of bacteria is more. And if you are using a different concentration, you can see here the number of bacteria when compared to control, the bacterial number is going down. So, by using this time dependent and concentration dependent study, we can confirm the antibacterial activity of your nanomaterial. An additional advantage of using this GFP E. coli is we can also do the fluorescent analysis for assessing the antibacterial activity. For example, in this case, when we are treating with the AZ-ZNO nanocomposite, you can see here this untreated E. coli. In untreated E. coli, this is giving a very high fluorescence, and with respect to concentration of nanocomposites, you can see that the fluorescent intensity is going down, and you can see that at high concentration it is completely gone down, fluorescence is completely gone down. And here we used only nanocomposites in the bacterial growth medium as a negative control without any bacteria. So, that is a negative control, and with respect to the concentration you can see that the fluorescence is going down. By using this fluorescent analysis also we can assess the antibacterial activity of nanomaterials. The next method is the disk diffusion method. So, this is a rapid and easy semi-quantitative method for assessing the antibacterial activity of diffusible antimicrobial agents from different drug delivery platforms. In this, we have to add 100 microliter of overnight grown culture, and you can use a sterile swab and you can spread it on the complete agar plate like this. Either you can use the sterile swab or you can use this kind of spreader and spread the bacteria completely by rotating the plate with bacterial culture. Then you have to place the disk of different sample. For example, you made some nanofiber membrane and you want to understand their antibacterial property. You can cut the nanofiber membrane into small circular disc, and that can be kept it. And in addition to that, you can keep like a negative control, that is only filter paper, and that is another one is positive control, that is a antibiotic disc. That means completely it will kill the bacteria. So, we can use one positive control and one negative control, and also we can use the sample. So, the positive control is antibiotic disc. So, antibiotic disc where we can see the clear zone of inhibition, and negative control is only filter paper or only nanofiber without any antibacterial agents. And third one is your test sample, nanofiber loaded with some antimicrobial agents. And after incubating the plate, you can observe the plate and measure the zone of inhibition for test sample. You can see here. So, this is your circular disc, and followed by that there is no growth in this region. So, that is a clear inhibition of growth. So, that is called zone of inhibition. For example, this is your sample, and in the surrounding there is no bacterial growth, and whereas here there is a bacterial growth. That means, So this is called zone of inhibition. This is called zone of inhibition. In this zone, it is not allowing the bacteria to grow. And by using this vernier caliper, we can also measure the centimeter of how much, or millimeter how much it is inhibiting the growth of bacteria, and we can plot it. So in this case, we used a silk fiber. This SSD is the silver sulfur diazine, and SS is the silk sericin, and SF is the silk fibroin. So this is a different nanofiber membrane. One is loaded with the antimicrobial agent, the other one is simple nanofiber. And you can see that when you are using the negative control, that is only filter paper. There is no zone of inhibition. And when you are using only scaffold, that is only nanofiber without any antibacterial agent, there also we are not able to observe any zone of inhibition. And when you are using the drug with a different concentration, you can see the clear zone of inhibition. Here you can clearly observe there is no growth. So that is called as zone of inhibition. So that can be measured and that can be also plotted to understand the antibacterial efficiency of the particular nanomaterial. And we can also use the various microscopic analyses to understand the morphology of bacteria which is undergoing the cell death. For example, we can use the atomic force microscope, and this is the untreated E. coli. You can see that it is a rod shaped bacteria, as I told earlier, and this is the Staphylococcus aureus untreated one. So you will get like this kind of bunch of grapes. The bacteria will look like this. And when you treat this E. coli and S. aureus, you can see that the bacteria's cell wall is damaged. That can be studied using atomic force microscopic, or we can use the SEM, scanning electron microscopic analysis. We can see that the bacterial cell wall is damaged by the nanocomposite. So as I told you in the electron microscopic lecture, if you want to confirm this damage is done by the nanocomposite, if you want to confirm that, what you can do, you can select this area and you can do the elemental analysis. And when you do the elemental analysis, if you are getting a peak for silver and zinc, that means, so the silver and zinc, it is attaching to the cell wall of bacteria and destroying the cell wall of bacteria. And we can also study the bacteria under the transmission electron microscope. So this is a single bacterial cell, that is the E. coli bacteria, and you can see here the bacterial cell wall is damaged by the nanoparticle. So we can confirm it when you are enlarging this and see that there is the black color dots. These black color dots are silver nanoparticle. So this confirms the presence of silver nanoparticles on the cell membrane of bacteria. We got the overall idea about how to assess the antimicrobial properties of nanomaterials. Let us see what is the mode of action of nanomaterials. The first one is, this is due to the electrostatic interaction. If you are using the nanoparticle with the positive zeta potentials, that means your nanoparticle is positive charge, and most of you know the bacteria and cell are negative charge. So through electrostatic interaction, this positive charge nanoparticle can easily attach to the negatively charged bacteria or cells, and it can attach to the cell wall of bacteria and it can damage the cell wall. Or the other mechanism is, it can also produce the ROS, that is the reactive oxygen species, and it can damage the bacterial cell wall. And as I told you, that DNA also is a negative charge. So this positively charged nanoparticle can easily attach to the negatively charged DNA, and it can inhibit the replication of the DNA and leads to cell death. Based on the various antibacterial studies and literature, numerous antibacterial mechanisms have been postulated. The first one is electrostatic binding. As I told earlier, the positive charge nanoparticles can easily bind to the negatively charged bacteria through electrostatic interaction. These metal nanoparticles induce surface oxidation, and the generation of reactive oxygen species enhances the oxidative stress, which leads to membrane disruption and kill the bacteria by damaging the cell wall. Once the cell wall is damaged, it leads to oozing out of damaged cellular material, These nanoparticles can also kill the bacteria by inhibiting DNA replication, transcription and translation process. The positive charge metal nanoparticles binds to the negative charge DNA and inhibit the DNA replication. It also binds to the ribosome and inhibit the protein synthesis, which leads to bacterial cell death. Hope you got the overall idea about antibacterial mechanism of metal nanoparticles. Let us see what are the various crucial factors affecting the antibacterial mechanisms of metal nanoparticles. The first one is nanoparticle size. As you know, the smaller nanoparticles have a greater surface area to volume ratio, which allows them to attach more easily to bacteria. The smaller the nanoparticle size, the stronger the antibacterial potential. Compared to the standard antibiotics, small size nanoparticles have higher antibacterial activity. The next one is nanoparticle shape. The shape of the nanoparticles can affect how it interacts with bacteria. For example, the spherical shaped nanoparticles have higher antibacterial activity compared to other shapes. The third one is nanoparticle surface charge. As I told earlier, the positively charged nanoparticles are more likely to interact with the negatively charged bacterial surfaces. And next one is capping agent. So, that depends on the type of capping agent used to stabilize the nanoparticles. So, that will also affect the antibacterial properties. For example, if you are using the PI, it will give positive charge to the nanoparticle. So, it can easily attach to the bacteria. And if you are coating the nanoparticle with PEG, so it has the negative charge. So, based on the your capping agent or the coating agent, and the antibacterial properties also will vary. And next one is environmental factors. The pH and ionic strength of the medium also affect the antibacterial activity of the nanoparticles. And suppose if the pH and ionic strength is not optimum, that leads to the aggregation of nanoparticle. Once the nanoparticle is aggregated, the interaction with the bacteria will be less, and that leads to less antibacterial activity. So, to achieve high antibacterial activity, we have to optimize the parameters. I hope you got the overall idea about how to assess the antibacterial properties of nanomaterials. Let us go to lab and learn this technique more in detail. In this experiment, we will learn how to assess the antibacterial properties of nanomaterials. To proceed with the experiment, we require nanomaterials. Today, we will first synthesize cellular nanoparticles. For this, we will use AgNO3 as a precursor, sodium borohydride as a reducing agent, and SDS, that is sodium dodecyl sulfate, as a capping agent. Additionally, we will need microtips, micropipettes, and ultra pure water, 2 microcentrifuge tubes, one for the control and another one for synthesizing cellular nanoparticles. First, we will add 1 ml of ultra pure water to both tubes. Next, we will add 3.34 microliter of 0.1 molar AgNO3 solution to the microcentrifuge tube. We will then add 2.66 microliter of freshly prepared 2 mg per ml sodium borohydride as a reducing agent to the microcentrifuge tube. While in the control tube, we will add the same amount of ultra pure water. Finally, we will add 90 microliter of 0.1 molar SDS as capping agent to both tubes. Take both tubes and shake them vigorously. Here you can observe that the tube where we added reducing agent turns pale yellow. This confirms the synthesis of solar nanoparticles, while the control tube remains transparent where we added only ultra pure water instead of reducing agent. Now we have synthesized solar nanoparticles. We will assess the antibacterial properties of solar nanoparticles using the disk diffusion method. The first step is to sterilize all the materials needed for the experiment under a laminar flow hood. We will begin by dipping a triangular glass spreader into ethanol, flaming it to sterilize, then shaking it to cool down. Place the glass spreader in a stand to allow it to cool down further. Next, we will add 5 microliter of the following samples to separate sterile discs, such as positive control, that is antibiotics, negative control, that is LB broth, and a test sample, that is solar nanoparticles, and allow the disc to dry under sterile conditions. Now we will add 100 microliter of overnight grown bacterial culture to a LB agar plate. Using the sterile triangular glass spreader, we will spread the bacterial culture evenly on the LB agar plate. After that, we will place the earlier prepared 3 disc positive control, negative control and solar nanoparticles on the plate. Now we are going to place the 3 discs on the plate with respect to its naming on the plate. Once it is done, place the negative control plate as well as the test plate in the incubator. After overnight incubation period, here we can observe zone of inhibition for our nanomaterial treated disc as well as the positive control disc which is having antibiotics. So today we learnt the synthesis of cellular nanoparticles and their evaluation for antibacterial properties using the disc diffusion method. Here we observe the antibacterial properties of cellular nanoparticles under normal light and UV transillumination. The cellular nanoparticles exhibit a zone of inhibition against E. coli bacteria, which is compared to the positive control, that is the antibiotics, and the negative control, so that is the only simple broth. So here you are not able to see any zone of inhibition, but in the test sample where the cellular nanoparticle is there, you can see the clear zone of inhibition, and in the positive control also we are able to see the clear zone of inhibition. Whereas in case of negative control, where we added only the broth, so there is no zone of inhibition. As a summary, in today's lecture we learnt various techniques to assess the antibacterial properties of nanomaterials, and we also learned the antibacterial mechanism, and through practical demonstration we learnt how to assess the antibacterial properties of nanomaterials. Thank you for your kind attention. I will see you in another interesting lecture.